Good morning, students. So, portions are completed and the practicals are almost done. Now we are moving on to revision part. So, we will start with the revision of the last chapter that was discussed, biotechnology and its applications. Okay. So, I will be covering some questions today and some questions tomorrow. By that we will try to wind up this chapter, revision part, and on the next week, Friday, you will be having your test on the same chapter. Okay. So, moving on with biotechnology and its application. After the school reopens, after the Christmas vacation, you will be having your terminal examination. For that, this chapter is also included. Okay. So, this chapter, biotechnology and its applications, revision we are going to start. Yeah, here we go. So, this time you know that new system has been introduced in the question pattern is changed and you are having assertion reason type of questions. You may be familiar with this because in physics and chemistry it was already there, but in biology newly introduced. Okay. So, we will discuss some questions, assertion reason question, followed by the rest of the questions, right. Now, moving on with the first question. Agrobacterium tumefacium is popular in genetics engineering because this bacterium is associated with the roots of all cereal and pulse crops. That is assertion. Okay, they are giving one assertion, one statement, and after giving the statement they are justifying it with a reason. What is the reason they are saying is that a gene incorporated in the bacterial mesosome gets automatically transferred to the crop with the bacterium is associated. You can look into it, isn't it? Now, the options are, if both assertion and reason are true and the reason is the correct explanation of assertion. Usually the options remains the same. They say that assertion is also correct, reason is also correct, and explanation is perfect. Then it is A option. B, both assertion and reason are true, but the reason is not correct. Okay, it's not the correct explanation for the statement. C, if assertion is true and the reason is false. Next, what they are saying, assertion is okay, the statement is correct, but the reason given is wrong. And the last one, what they are saying is that both are wrong. The assertion, the statement is also wrong, and the reason for the given statement is also wrong. So, answer for this is D, both assertion as well as reasons are false. Listen why, because Agrobacterium tumefacium, what do is that they are causing a Crohn gall disease is the disease which is caused, and it is caused where? It is caused in your broad-leaved plants. It is not in the cereals and pulses, broad-leaved dicot plants only it is causing. So, that is why you can find that the assertion is wrong. Okay. So, mostly in the rose family it is causing, rose family, rose you know, isn't it? But whether the agrobacterium, what they have said, they are introduced in the mesosome, gets automatically transferred, was the reason given, that is also wrong. Where we are transferring this to the plasmid, isn't it? Extra chromosomal DNA. In the laboratory, we are moving this sort of gene into plant, and that microbe, that is the bacteria, is introduced into a plant for investigatory process for genetics. So, it is not introduced into the mesosome, it is introduced into the, genes are introduced where? Into the plasmid. Okay. And then that plasmid is introduced into the bacterium, agrobacterium, and then it is going and infecting the dicot plants, broad-leaved plants. Okay. So, both the assertion and the reason here is wrong. Going on with the second question, GM crops can affect human health by causing allergic reaction. Sometimes what happens is that GM crops is affecting the human health by causing allergic reaction. For that, the reason what they said for the statement is that transgenes in commercial crop can endanger native genes. Okay. So, transgenes are endangering native species. Like for example, Bt toxin gene expressed in pollen. So, Bt toxin, if it is expressed in the pollen grains, if this Bt toxin is there, the honeybees, those who are coming and visiting, can get affected. So, that is what they are saying. Okay. So, here again, what and all, both assertion and reason are true, and the reason is the correct explanation of assertion. No, you can find both assertion and reasons are true. If pollen grains are having Bt toxin, it can affect honeybee, and thereafter you can find what is the assertion given, GM crops is affecting some of the human population, causing allergic reaction. But the reason is not for humans, it is for honeybees, right. So the reason is wrong, but both are correct. As such, when you are saying assertion statement is also correct, GM is causing some allergic reaction, and the reason is also correct. That is, the reason statement is also correct. What is the reason? The reason what they have given the statement is that they are affecting the honeybees. But whether the reason is the correct explanation of the assertion? No. They are not saying about human beings. They are saying about some other things, honeybees. That's why assertion and reasons are true, but reason, when you say, it's not the correct explanation of assertion, right? So that is the answer for the second question. Moving on with the third question. Flash saver tomato was the first transgenic commercial crop that entered the market. Okay, yes, so that is number 1. Roundup variety of soya bean was prepared through breeding. So two different things, you see, one is they are saying something about soya bean and other one is saying something about what is that, tomato. Okay, so as such, when you are saying what will be the answer is C, if the assertion is true but the reason is false. When you are saying the assertion is true, okay, so the second reason is wrong, assertion is true. So what was the reason, what was given? The reason is soya bean, Roundup variety of soya bean was prepared through breeding. That is wrong. The first assertion is correct. Flash saver tomato was the first transgenic commercial crop that entered the market. But when you are saying, the reason is not the explanation for assertion. So the answer will be C. What is it? If assertion is true but the reason is false. Clear? Yeah. Now moving on to the fourth question. A gene from Bacillus thuringiensis is incorporated in the plant genome to increase the yield. Yeah, Bacillus thuringiensis, you know, they are acting as what? Pesticides, isn't it? So obviously if the pests are not affecting, then the yield will be more. Reason for the same is that the Bacillus thuringiensis has Bt toxin which kills the larvae of the insects. So whether both are correct or not? Yes, both are correct. Bacillus thuringiensis is increasing the yield when I am incorporating in the plant, as they are acting as pesticides. Reason is also the correct. It is having a Bt toxin which kills the larvae. So what will be the answer for this? Answer will be A, if both the reason and assertions are correct, and the reason is the correct explanation of the assertion, right? Now moving on to the fifth question. Rosie, the first transgenic cow, produced human enriched milk. You know, isn't this alpha lactalbumin we have learnt. So Rosie is the first transgenic cow, yes, and it produced human enriched protein milk than normal cow. This is having human enriched protein milk. So assertion is correct. Reason, the milk contained the human alpha lactalbumin and was more balanced product for babies than the natural milk. This is also sounding correct, right? So the answer will be both assertion and reason is correct. Enriched milk alpha lactalbumin is present in Rosie, which is comparatively good compared to the normal cow's milk. Okay. So with this, assertion reason questions are done. Next we are moving on to some MCQs. Okay. So the first one is, Bt cotton is not, see, Bt cotton is a GM plant, yes or no? Yes. It is insect resistant, yes. A bacterial gene expressing system, yes. Bt is Bacillus thuringiensis, it is a bacteria. So A, B, C is correct. Is not resistant to all pesticides is the answer. Answer will be D. It is not resistant to all pesticides. It is a pesticides. They are not resistant to pesticides. Actually, when you are saying this Bt is itself is a pesticide. So the answer which is not applicable to Bt will be what? Resistant to all pesticides. So the answer will be number D. So Bt is a GM plant, is insect resistant. Thereafter, when you are seeing, it is causing what? It is a bacterial gene expression system. So the answer will be yours. It is resistant. It is not resistant to all pesticides. Now the next question is, C peptide of human insulin is a part of mature molecule, insulin molecule, responsible for formation of disulfide bridge removed during maturation of proinsulin to insulin, responsible for its biochemical activity. So mostly answer will be what? It will be C. You know that C peptide. So the proinsulin will be having what? The proinsulin will be having a C chain. So during maturation, what we are doing? We are removing this proinsulin C chain is being removed. So, C peptide of human insulin is removed during the maturation of proinsulin 2 insulin will be your answer. Moving on to GEAC, what is the full form? Direct, you can write Genetic Engineering Approval Committee. So, the answer is C. Alpha 1 antitrypsin we discussed, it is used for treating what? A respiratory disorder, emphysema. So, the answer will be D. A probe is a molecule, you know what is probe used for? Autoradiography, isn't it? So, it will be having a single stranded DNA or a single stranded RNA with a radioactive substance, so that we can diagnose the disease or diagnose the defective gene. So, a probe, which is a molecule used to locate specific sequence of mixture of DNA or RNA molecule, could be C is the answer. It can be either a DNA or a RNA single stranded with a radioactive molecule, which is called as what? A probe. Okay. So, the answer will be C. It can either be a single stranded RNA or single stranded DNA, and it is tagged with a radioactive molecule called probe. Now, sixth question, choose the correct option regarding retrovirus. So, retrovirus, you know, it is a RNA virus, isn't it? And it gets converted into DNA when it is infecting, when it is getting into our body like HIV. They get converted into DNA by a process called as reverse transcription, which enzyme reverse transcriptase is playing a major role. So, the answer will be A. Retrovirus is an RNA virus that can synthesize DNA during infection. The next is the site of production of ADA, adenosine deaminase, isn't it? So, you know where is the site? It is WBC, is lymphocyte. So, where is ADA produced in the body? In lymphocytes. So, answer will be B. The site of production of ADA in the human body is lymphocytes. Next is your a protoxin. You know protoxin is an inactive form. So, a protoxin is a primitive toxin, a denatured toxin, toxin produced by protozoa, inactive toxin. You know it is an inactive toxin. When it gets into the gut, you can find that alkaline pH of the gut of the larvae causes what? Activates the inactive toxin. So, the answer will be D. It is an inactive toxin. Moving on with the next question. Pathophysiology, what it is? It is a study of altered physiology of host. Physiology means what? Functions. Okay. So, pathophysiology is because of an entry of a pathogen, if some disease we are getting, how our host is getting altered because of that pathogen, that study we call it as what? Pathophysiology. Okay. So, pathophysiology is the study of altered physiology of host. I was having my own functions, now my functions are altered because of the entry of a pathogen. They have changed my function, altered my function. So, that one we call it as what? Pathophysiology, the study of what is that? Altered physiology of host. Moving on with the next question. What is the next question is the trigger for activation of toxin of Bacillus thuringiensis, you know it is the alkaline pH of the gut. So, the answer will be C, alkaline pH of the gut, right. Golden rice is, you know, it is a rice which is rich in what? Beta carotene. Okay. So, beta carotene is more in this golden rice. So, answer will be again C, a transgenic rice. Yes, it is transgenic rice produced by it is a GM plant having gene for what? Beta carotene introduced into the normal rice. Now, in RNAi means what? RNA interference. Genes are silenced using, how we are silencing the gene? You know defense mechanism in eukaryotes, what is it? They are producing double stranded RNA, dsRNA, which go and suppress the mRNA. So, that is why the answer will be RNAi is a interference, RNAi or RNA interference takes place in all eukaryotic organism. It is a cellular defense mechanism, and this method involves silencing of specific mRNA due to complementary dsRNA. So, again the answer is what? The answer is dsRNA, which is suppressing the translation of the mRNA to protein. Okay. So, this is also similar. So, you know dsRNA is the defense mechanism. So, the answer will be C. Moving on to the next. First clinical gene therapy was done for the treatment of, for what treatment? You know ADA, isn't it? So, the answer will be SCID. So, severe combined immunodeficiency, 4-year-old girl, you know, isn't it? So, this is done, you can learn. ADA is an enzyme which is deficient in the genetic disorder SCID. What is the full form for ADA? Adenosine deaminase. So, you have learned this one, hope that is clear, nothing to explain. Silencing of a gene could be achieved through the use of RNAi only, antisense RNA only, by both or none of the above. So, listen students, antisense term you have learnt in this RNAi, isn't it? So, RNAi as well as antisense RNA both can be used for silencing of a gene. So, the answer will be C, by both RNAi as well as antisense RNA. Next we are moving on to very short question. Okay. In view of current food crisis, it is said that we need another green revolution. Tell the limitations regarding green revolution. See, what are the limitations? So, the first limitation is that this fertilizers, pesticides, green revolution, you know, they are using what? Chemical fertilizers and pesticides. So, this chemical fertilizers and pesticides, one is that it is very too expensive for the farmers to get it, and this pesticides, when you are seeing, after introducing it into the ecosystem, into the soil, what happens is that when this pesticides gets washed off in rain and reaches the pond, it causes what? Eutrophication. So, what it is causing? It is causing eutrophication, and it is also causing what? Water logging, as well as when the water is getting logged. Imagine the water, because of rain, the water is getting logged in some places because there were not proper channels. Okay. So, when water gets logged over there, you can find they are getting evaporated after some time, and all the salts are remaining on the soil, and the soil is turning saline. Okay. So, like that, if saline soil will not help in the growth of the plant. So, like that it becomes affecting the, which one? The ecosystem is getting affected. So, that are the limitations of what? Green revolution. So, we are not going into green revolutions, right. Expand GMO, how it is different from hybrid. You know what is GMO, genetically modified organisms. How it is different from hybrid? Hybrid, you know, hybrid, when we are producing an hybrid, see, listen students. So, genetically modified organism, how? So, this is a, imagine this is a, what is that? A cell. Into the cell I am introducing the bacteria which is containing my foreign gene. That is how I am producing what? A genetically modified organism. But in hybrid, how it is done? I am selecting two parents. One is high yielding and another one is disease resistance. Now, they two are pollen grains from one is introduced into the stigma of the other, and they are producing a F1 generation. It is an hybrid which is having a combination of high yielding as well as disease resistant. So, this one we call it as what? Hybrid. And how it is different? See, GMO, when you are seeing correctly what gene I need, that can be introduced into the plant. But imagine this is high yielding, but it is having some defective characters, bad characters. Sometimes in that hybrid also you can find the bad characters are getting expressed. So, correctly what I want I may not get in a hybrid, but in the GMO correctly what I want I can get it, because I will introduce only those genes which I want and I will remove those genes which I do not want. That is why you can find the hybrid is different from GMO, because it is differ from hybrid, because hybrid cross is done between a total genome of two species. Totally the genomes are getting crossed over and a new species is formed. Okay. You cannot particularly select and do it, but when you are seeing extra chromosomal entity, so when you are moving into this GMO, it is an extra chromosomal entity. Okay. So, correctly what gene I want can be introduced. So, that is the difference between GMO and an hybrid. Differentiate between diagnostic and therapeutics. Listen carefully, what is diagnostic and what is therapeutics? Diagnostic is that you are going to diagnose whether you are suffering from a disease or not. That is called as diagnostic. Okay, just to find out whether I am having a disease or not. That is called as diagnostic. And what is therapeutic means? Therapeutic means I have to treat disease, therapy. So, I am now having a disease, I know that I am having a disease. Now, how to treat that disease? That is therapeutics. So, diagnostic is to find out whether I am having a disease, therapeutic is to treat the disease, how to treat the disease. Okay, the ways to treat the disease. So, diagnostic, for example, ELISA test is done to confirm whether I am having HIV or not. So, that is diagnostic. What is therapeutics? Antibiotics. So, I am having a bacterial disease, so I am going to take medicine. Antibiotics I am going to take so that I can get cured, treated from the disease. Okay. So, that is therapeutic. Clear? Examples I have given. Moving on to next one. Give the full form of ELISA. Which disease can be detected using it? ELISA, you know, enzyme linked immunosorbent assay. Which disease can be discussed? We have discussed AIDS, isn't it? Principle underlying. What is the principle underlying? Antigen antibody interaction. So, by just checking whether the antigen is present in my body or antibodies are produced against the antigen. So, antigen is introduced, because of which if my body is having those antibodies, which means that that germ is there in my body, that is why only the antibodies are produced. So, like that I can confirm that I am having the disease by the presence of antigen or antibodies interaction, how it is going in my body from a blood sample. We can test and we can confirm whether we are having AIDS or not. Can a disease be detected before its symptom appears? Yes, we can detect it, isn't it? How can we detect it? I told you, when a pathogen is entering into a body, in the beginning when you are saying, like for example, you take HIV itself. In the beginning itself, I cannot understand whether I am having a disease or not, because maybe my immune system is so strong. Okay. So, sometimes what happens is that the disease after entering into my body remains suppressed, they are not multiplying. But gradually what will happen, after some days my immune system will become weak, this pathogen will weaken my immune system and I will be getting some symptoms. But by the symptoms it is coming in my body, I would have what happened, I cannot treat it because it would have reached the advanced stage. So, in order to see whether I am having, if there is any doubts or someone else is suffering, so that I am likely to suffer, I am having hereditary disorders or something like that, if you think, you can go for yearly diagnosis. How you can yearly diagnose whether you are having a disease or not? By using PCR. Even as one single gene is present, a foreign pathogen is present, that can be detected with the help of PCR. Okay. So, yearly diagnosis can be done with the help of a technology called as what? PCR, polymerase chain reaction. Okay. So, in that I am amplifying the gene, amplifying the pathogen, and I will then learn the pathogen, what the pathogen is, and then I can understand if this pathogen is present, then automatically I am suffering from the disease. Okay. The next is your write short notes on biopiracy, highlighting the exploitation of developing countries by developed countries. You know, biopiracy, patenting, isn't it? So, developing countries are being exploited by underdeveloped countries. Okay. So, developing countries like India, how we are exploited? We know that we are having much resources, we do have many traditional knowledge about the same. So, developed countries are visiting our places, taking all the information from us, and they are developing those varieties. After taking the information from us, they are using those information for making new items, and they are getting the patent for it, and then they are selling it in their market, as well as we are importing it from their country. Our own resources are being imported, our own resources are being taken. So, we are exploited in that way. Okay. So, that just go through what has been given in a book, that is more than enough. Now, many proteins are secreted in their inactive form. This is also use of many toxic proteins produced by microorganism. Explain how the mechanism is useful for the organism producing the toxin. Now, listen students, what they are saying, that inactive form of toxins are produced by this bacteria. So, like Bacillus thuringiensis, they are producing a toxin Bt, but the toxin Bt is not causing harm to bacteria. Whether it is causing any harm to the bacteria? No, it is not going to cause any harm to bacteria, because it is in inactive form. So, the bacteria can survive the toxin, they are not having any problem, but it can be advantageous for us. How? This inactive toxin, when it is moving on to the intestine of the gut of the insects, it is getting converted into what? An active form. So, pH, temperature helps in the conversion of inactive toxin into toxin, and the insect is killed. But the bacteria, when you are seeing it, for them it is an advantageous, it is good for them, okay, because they are not killed by the change in the thing, they are having the mechanisms to withstand the toxin. So, it is useful for the bacteria, but harmful for the insects, and we can use the same principle for developing insect resistant varieties. Okay. Yeah, while creating genetically modified organism, genetic barriers are not respected. How can this be dangerous? So, nowadays everyone is researching, not everyone, many research people are researching to develop many GM crops, isn't it? GM organisms, maybe animals also. What is the danger effect? I told you. Sometimes what happens is that when I am introducing this gene into an organism, sometimes that gene can work out in a negative impact, means they can give rise to some other forms which can be dangerous to the ecosystem. It can cause some dangerous deteriorating effect which can kill the normal varieties. Okay. So, like that the ecosystem can get affected. So, genetic barriers, if you are not respecting it, means if it is not fulfilled, genetic barriers are there, you have to get approval from the committee, thereafter only after the approval, maintaining all ethical issues, only you have to go for the process. Just like that, randomly if you are going for, then sometimes it may give a very dangerous effect which can even cause the death of the human race as well. So, we have to be very careful when we are developing genetic modified organism. Why has the Indian Parliament cleared the second amendment of country's patent bill? You know, Indian Parliament has recently cleared the second amendment, wherein they are taking all the considerations regarding patent terms, everything is being taken care. They are giving emergency provision, research, developing activities and all, they are giving patents so that the Indian people can develop their Indian varieties and get the patent for their own varieties, so that the other countries are not taking advantage of our resource. Okay. So, for that only they have cleared a bill wherein they are means clearing all the doubts, all clarifications regarding what? Regarding patents. Okay. Give any two reason why the patent on Basmati should not have gone to American company. You know, Basmati, it is our tradition. In Indian folk songs, even in the poems and all, you can find this term Basmati being used, and it is an Indian variety. So, this Indian variety, the company, American company, they have no rights to get patent for the same, because we are the one who is using this from thousands of years ago. So, the American company have no rights to get patent for the same. Okay. So, that is the answer. So, rice is important food grain, you know, in Asia, and we are producing around 200,000 varieties of rice. Diversity in India is one of the richest in the world, you know. Basmati is distinct in aroma, smell and flavor also, and there are about 27 documented varieties of Basmati we already have. So, this is reference, this Basmati is also referred in our ancient textbook, folklore and poetry. So, no American companies has rights to take patent for the same, the Basmati which belongs to India. That is the answer. How was insulin obtained before the advent of rDNA technology? So, in rDNA technology, you know, C chain is removed and disulfide bond is made, and we are getting combining the A and the B chain, and human insulin is produced, isn't it? So, that is through rDNA technology. Before that, how the insulin was obtained? You know, from slaughtered cattle and pigs, from there the insulin was taken and it was used. But what was the problem with that? They were causing some allergic reactions, isn't it? Since it is not equivalent to human insulin, they were causing some allergic reaction. That is why only we developed what? rDNA technology for developing insulin nowadays. With respect to understanding diseases, discuss the importance of transgenic animal models. So, yeah. So, understanding diseases, you know, in order to find out how the disease is developed, what are the symptoms. So, for that we are developing what? Transgenic animals. Into them we are introducing the disease causing gene, and thereafter we are learning, investigating the disease, how the symptoms are appearing, Then after finding out that these are the symptoms, then we are trying on that animal, giving different type of treatments and how the treatment can be effective, whether the treatment is working out or not, whether the treatment is positive and the animal, transgenic animal, is cured. So like that we can test and thereafter we can use it in our human population. So we are developing this transgenic animal models so that I am introducing the pathogen or the gene which is causing the disease into the transgenic organism. I am studying the symptoms which are developed and I am developing some technologies to treat the diseases, and I am testing in that transgenic organism whether the disease has been treated or not, and then we are using it in our human population. So this is how we can understand the disease and help in developing many treatment. I cannot use a human being for the same, isn't it? If I am using the human being can harm the human being, right? So in the transgenic organisms we are using the same, especially transgenic mice. The name the first transgenic cow, you know, it is Rosie. Which gene was introduced? Alpha lactalbumin, you know. PCR is a tool for early diagnosing of, yeah, you know, PCR. Why it is a tool for early diagnosis? Because even if a small sample, even if very few sample of any pathogen is present, that can be easily amplified using what? PCR, and we can learn what is that gene, what is the pathogen, how it is causing the disease. Okay. What is GEAC? You know, it is Genetic Engineer Approval Community. And what is its objective? Examine the validity of GM. GM is okay, it is not having any deleterious effect or deteriorative effect. That one we have to, this committee is examining. And thereafter what they are doing is that inspecting the safety of introducing GM for public services. First they will inspect, everything is okay, then only they will allow to release it in the field or in the market. Okay. For which variety of Indian rice the patent for was filed by US company, you know, Indian Basmati semi-dark variety, isn't it? US company, our Basmati variety with their one variety, they mixed it and they produced semi-dark variety of Basmati. Discuss the advantage of GMO. GMO advantages in the first part itself we have discussed. GMO is tolerant to abiotic stresses. For crops, when you are saying they are not affected by cold, drought, salt or heat, is not a problem, they can grow. If it is too cold, if it is, there is no water, or if it is having much salt or it is too hot, they all can grow. Okay. They are not dependent on water, they are not dependent on pesticides, and they are reducing our post harvest losses, because most of the products which are obtained are in good form, so that post harvest losses are reduced. Increased efficiency of mineral usage by plants, correctly they are using the minerals, they are not exploiting it. Okay. And the last is nutritional enriched varieties can also be obtained, like vitamin A enriched rice can be obtained with the help of what? This technology. Okay. So GMOs can help in making all these things. Modified organisms are having them. With this I am stopping for today. In the next class we will be discussing about the short answer type questions of the same. Okay. Thank you.